Plant nitrogen status plummets under carbon dioxide enrichment. Shoot nitrogen and protein concentrations in C3 plants fall 17 and 49% respectively at elevated carbon dioxide concentrations. These declines are similar in plants receiving different levels of nitrogen fertilization and are much more than double what we would expect if this were simply a case of dilution through growth. Were it dilution through growth, the additional biomass that C3 plants accumulate under carbon dioxide enrichment would decrease shoot nitrogen or protein by only 8%. Consequently, exposure to elevated carbon dioxide seems to inhibit some aspect of plant nitrogen metabolism. We recently discovered a key interaction between carbon dioxide metabolism and nitrogen metabolism in plants that accounts for many other responses to carbon dioxide enrichment. Contrary to popular opinion, photorespiration is not just a process that consumes sugars and generates waste heat. It is not just the unfortunate vestige of plants evolving when atmospheric carbon dioxide concentrations were much higher than they are today. In a wide range of C3 species, conditions that inhibit photorespiration, namely elevated carbon dioxide or low oxygen atmospheres, also inhibit shoot nitrate assimilation into amino acids. Photorespiration appears to provide some of the energy required to convert nitrate to ammonium. This coupling of photorespiration to nitrate assimilation makes C3 species more energy efficient than previously thought and explains why more than 95% of Earth's flora is composed of C3, not C4 species. This discovery provides a simple explanation for the variation in plant responses to CO2 enrichment. Plants differ in their relative dependence upon ammonium and nitrate as nitrogen sources. Variation in the response of C3 plants to elevated carbon dioxide concentrations derive from variations in nitrogen sources. For loblolly pine, plants at ambient carbon dioxide concentrations grew similarly under ammonium and nitrate nutrition. But plants at elevated carbon dioxide concentrations grew much more slowly when their sole nitrogen source was nitrate than when it was ammonium. The phenomenon of carbon dioxide acclimation derives from the decline in plant enzymes required for photosynthesis and growth that develops with exposure to elevated carbon dioxide concentrations, because elevated carbon dioxide concentrations inhibit nitrate assimilation into proteins, and therefore protein synthesis declines.